So thank you, everyone. Thank you, Nicole. So what you have heard from Nicole and Annie is really why metabolomics and really understanding the intricate aspects of using the data to find those very nice patterns within the biology, right? And so what I'll show today is just how we do that, how we do untargeted metabolomics at Metabolon. And then also, how do you take that data and to Nicole's example, identifying this mystery metabolite being quite important in your data, and if that is a mystery metabolite or if that is a known metabolite, how do we go about going more in a targeted fashion to validate that in a larger cohort and start tracking those metabolites of interest in a given cohort? So that's the big overview of what I'll be presenting, and then at the end I'll just round it up with an example where we help the client specifically do that, go from a global analysis all the way to a targeted, and even in a clinical setting at the end of the day. So this is really why I wanted to also preempt this slide. Is really where is metabolomics data being used, and where do we see it as being very informative? So I'll just quickly get a pointer here. So when we think about the clinical pipeline as we have it at the moment, or drug development pipeline, we start on the far left side, really with the discovery, exploratory approach that Nicole and Annie talked about quite a bit here. And so that is very much, we want to look as wide as possible at as many different metabolites and be able to use that big data and connect it to other clinical data or other omics data. But now we're starting to get a better picture of what is happening within this biology, and as we progress across this drug development pipeline, this is where we want to not necessarily have the lens open quite wide, and we start wanting to focus on certain chemical groups or specific metabolites to be able to now actually go and quantify those with absolute quantifiable targeted panels. And so as in the bottom part, I'm also showing just that how it links up with some of the products we have at Metabolon. In the exploratory side, this is the Global Discovery Panel that Nicole referred to as the HD4 panel. It's a whole other name for that. And then as you progress to the more targeted approach, we have several off-the-shelf targeted panels, but we also have the option of customizing that. And just to that point, so we have the Global Discovery Panel on the left-hand side. We have now recently launched a few focused discovery panels that have a reduced number of metabolites, but very specific to liver function, kidney function, as well as biological stress. So being able to profile those all quite nicely. And as we progress to the right-hand side, this is then becoming more targeted. We have our complex lipid discovery panel, as well as several other smaller targeted panels. These give you absolute concentrations, and then we can even go to single analyte panels as well. So a little bit more about our discovery panel, or the HD4 untargeted metabolomics panel. And so this is really a front-to-end service that we provide. We have we usually engage with our clients on a very early stage where we do study design. We make sure that the samples that were collected or are going to be collected will be the best for a metabolomic study. We think about power of the study. We take all of that into consideration with our clients and advise them correctly on that, and then we can progress to the next step, which is really receiving those samples pre-processed, and we would then at Metabolon process these for metabolomics purposes, and then that goes onto our platform. So our platform consists of 4 different arms, and so this is really allows us to have that wide chemical diversity measurement that we provide. And so in a given plasma sample, this can be anything from 1,100 to 1,500 metabolites that we do identify, knowns and unknowns. And so then that all of that information that we gather on those 4 arms, really, we pull that all together, and that goes then into our curation platform, where we match that against our database. And so our database is a little bit different than a lot of other providers in the sense is that it's a database that has been built on authentic reference standards. So when we match against this database, we use 3 different criteria to match against it, but it matches it against data collected on these reference standards run on the same platform as what the samples will be run. And so that really allows us at the end of the day to get that confident identification. And I'll go a little bit more in the next slide into that. But then at the end, really having this quality data, we also have the option of providing some interpretation from a metabolomic standpoint. And so this is also something where we add a lot of value to our clients, is that they don't have to know metabolomics. We are the experts on metabolomics, and this is where we can support our clients in providing insights from a metabolomic standpoint, and then that can then of course become incorporated with other data as well. So back to the confidence, and this is really a fundamental aspect at Metabolon. Is firstly, validation is very important to us. So matrix validation, metabolite validation, and then at the end of the day, really being able to provide on an annotation basis, what you see in this pyramid represented is that we move towards the top level, the level 1 for most of our identifications. And so that really provides you that insight, that confidence that if we say it's glucose or galactose or galactose, is really being able to say that they are, in many cases, look the same on a mass spec, but we separate them from each other, and we can really authentically tell you that that has been matched against the standard. And so you can take that to the bank. On the right-hand side is another part of our philosophy. It's just not to give you a list of metabolites and say, there you go, figure out where does that fit into the biology. It's really each of the metabolites that we identify is immediately put into a subcategory and a supercategory of, on the basis of chemical ontology, to say where do they belong, right? So these are just on the right-hand side some examples of the pathways that we cover. And so within this global discovery panel that we have, so really giving you a vast array of metabolites covering 70 different biological pathways that we can profile. And so this then gives you a lot of opportunity to discover those correlations with genomics, transcriptomics, and proteomics. And then once again, to that last part of the pipeline that we of our service, is really that we provide a comprehensive interpretation service as well and support. So not only do we provide the data, which is the intensity data, normalized data that we provide our clients, but also we put that in a nice heat map if we do the statistical analysis, so that we can put both biological contents and statistics into one view. And then on the right-hand side, we also have some nice pathway mapping tools available on our portal that allow you to get that biological, the pathway context as well of these metabolites. And in the middle is really the report written up by our PhD scientists that take into account all the data that we are providing. That can also include metadata from our clients. So if there are specific clinical trends that they're aware of, genomics, proteomics trends, even though we don't analyze that, we can use those trends to inform our interpretation of the metabolomics data. So that's very much our global discovery panel. It's really quite an exhaustive service that we provide from a metabolomics standpoint, and we can really do that depending on the need of our clients. We can provide it at all those different levels. And then when it comes to targeted metabolomics, so the one big targeted panel that we have is called our complex lipid targeted panel. And so even though this is quantitative, it is quite broad still. And so we cover 1,100 lipids in this panel. And so in itself, lipids are quite an interesting and complex group of metabolites, or lipids in this case. They have that inherent problem that they do overlap because they have very similar chemistry. So on a mass spec, they behave very similar, a lot of these classes. So they interfere with each other. So that is one of the biggest challenges within lipidomics. And then, of course, they also have a combinatorial complexity that we have to take into account. And so with our analysis, what we do quite uniquely is that we use ion mobility separation. So within the gas phase, we separate the different lipids from each other, the lipid classes from each other. And then after we separate them from each other, we do the speciation. So that it really allows us to remove any interference, but then also allows us to really do a deep dive into the species, the different species within each class. And then an additional aspect that we have introduced to our lipidomics analysis is really the aspect of quantification of such a large set of lipids. And so we have a set of 50 internal standards that we inject with each analysis. And so that allows us to not, those 50 internal standards cover the 14 lipid classes that you are seeing, but not only a standard per class, but we also take into consideration within a class the chain lengths, the carbon chain lengths, as well as the saturation levels. So we have representative internal standards to cover that as well. And so that really allows us to have a quantitative output of up to 1,100 or a little bit more than 1,100 lipids, covering these 14 classes. And then really to round that off quite nicely is that we also have a pathway mapping tool for lipids as well. And so this is very unique. Before joining Metabolon, I have never seen a pathway mapping tool for lipids because lipids are not very promiscuous. And so you can see that it's a different approach to normal pathway mapping tools. And so in lipids, you need to be able to visualize the pools of fatty acyl chains that are available to be incorporated onto these head groups in all of these the different classes. So this tool really provides that insight. It gives you also the option of doing discriminatory analysis within it and playing around with the statistical view. So a very nice tool to use to delve deeper into your biology. And so to another set of targeted panels that we have, and this is now quite a reduced number of metabolites, usually in order of 5 to maybe 20 metabolites that we have as panels, but some of them even go to 61 metabolites. And so this really fits very nicely into the pathway that I showed in this manner represented in the slide. So really, we use the global discovery panel to search for these unique metabolites or sets of metabolites that are indicative of the biology. After that, our clients usually want us to go a little bit further, and now taking that set of biomarkers that we have identified and actually go and validate them as a set on its own in a targeted fashion. And so at that point, we go and validate them both biologically and analytically with our clients in a research use only setting. We validate that, and when we start seeing that these are reproducible signatures in a quantitative manner, that then becomes the circle that we have here, that it can then go several iterations until we then get to a set of biomarkers that can be clinically validated as well. And so that really allows us to support our clients, depending on their need, really all the way to a clinical trial set of biomarkers that are fully validated. And we have done this also in GCP for several GCP panels as well, allowing our clients to use those. So here's the set of targeted panels that we have currently, what we call off the shelf. So these are ready to be run with taking into consideration the validated matrices. These are ready to be run tomorrow. And so quite a nice set of different types of biology that we cover. The ones that I've emboldened there, those are ones that we added this year to our repertoire of targeted panels. And so they're a little bit different in the senses that we have moved to a targeted panel style that doesn't only focus on chemical ontology, so covering one class of similar metabolites, but starting to look at different pathways and incorporating that into one panel. So really being able to have a biological focus of the targeted panels. And so just to give you 2 examples of this is the one that we know is the diabetes research market targeted panel. And as you can see through this list of these metabolites covered on here, some amino acids, some hydroxy organic acids, and even a lipid on here as well. So covering quite a nice diversity of biology in one panel and really giving insights into what has happened on a diabetes level. This is an 8-point calibration curve for each of these metabolites, really allowing you to get that quantitative data for, especially when you're looking into diabetes research. And the additional one, the last one for today, is a sphingolipid panel that we have as well. And so this is runs in a similar fashion as our complex lipid panel, targeted panel, but mainly focusing on the sphingomyelin pathways. And so really allowing you to look at these 5 different lipid classes and getting the quantitative result from this. This is really in cardiovascular disease seen to be quite applicable, where the ceramides, and specifically ceramide ratios, become quite informative regarding severity and predictability regarding cardiovascular events. So this really provides quite a broad set of ceramides to be able to calculate those risk factors as well. Quite a nice panel, and it falls within the suite, our inflammation suite. So most of the panels that came out this year were launched under our umbrella inflammation suite of panels that we have. And once again, I mentioned earlier Metabolon's philosophy regarding validation. For our samples, firstly, we don't only validate it for the metabolites, we also validate it for each matrix that a client might want to test. And so metabolites behave differently in different matrices. So for a proper validated study, we have to validate it both for metabolites as well as matrix. And so we have different levels of validation as well. So depending on the need, again, exploratory, non-GCP, or what we call RUO, and GCP as well. So adhering to all those different levels as required for these validation standards. And this is all done under certain, quite a few certifications and accreditations. Quite important, especially when you're working with human samples. We adhere to all of these and provide the necessary quality of data to be able to offer this. And so that was, in short, what we offer from Metabolon, both for the untargeted as well as the targeted panels that we have. So really allowing the full support of our clients from the start to the end, and however that is associated with other data, we can support that as well. And so with that, I'll move into a last case study just to give a real world example of how we did this. And it's not the only one, we do this regularly with our clients. It's really to take it from global all the way to targeted. So this was, as Annie already mentioned, this is a microbial metabolite case study where the client of ours was very interested in understanding what is impacting autism spectrum disorder, and understanding that disease a little bit better, but then also how can we treat it at the end of the day if we identify the causative aspect to it. And so as we know, autism spectrum is a group of heterogeneous neurodevelopment conditions characterized by social communication as well as some interaction, lack of interaction and so forth. So it's very difficult to test it. There are no real biomarkers associated with autism spectrum because the etiology is still very complex and we don't know everything of that. And so many affected individuals do also have a gastrointestinal dysfunction associated with that, together with other comorbidities. And so this aspect that there was a GI component to it, of course, raised the concern or the thought of might there be a microbiome aspect involved here as well. Of course, no drugs currently available to treat any symptoms regarding ASD. A lot of things in the works, especially FMT treatment as well. Fecal matter transplant is being considered by other companies. But in this example, there was clearly, it is well known that there's a genetic component, but also a microbiome component now, where we are today. And so now being able to understand this environmental component, which is the microbiome to some extent, can we actually measure that and understand that a little bit better? And so from studies, and I'll go into that example, so in a landmark study in 2013, where this company worked with Metabolon, they made the first connection between behavioral abnormalities linking ASD to decreased gut barrier integrity and modifications of the gut microbiome. And so the study implicated gut microbial metabolites specifically, and the very specific metabolite was actually the 4-ethyl phenyl sulfate, the 4-EPS molecule, showing that an increase in that metabolite was linked to increased behavioral issues, as conditions, and so forth, and some neurological development traits as well. And so in a follow-up study, they looked at, once again, at autism spectrum disorder individuals, as well as normal developing children. And once again, the 4-EPS molecule was very prominent in that cohort for the ASD patients having elevated levels. So in a subsequent preclinical development study, the same group showed that 4-EPS enters the brain and modulates neural activity and functional connectivity within the brain networks, underlying the emotional regulation as well. So they found that 4-EPS influences oligodendrocyte maturation and function. And so remarkably, pharmacological treatment that they then applied, and the clemenstein fumarate showed a correction of that to some extent. They then went and tested their specific product, AB 2004, and administered that to a cohort. Sorry, this was in a mouse model. And they saw that that was lowering 4-EPS levels in these mice. And so really also saw an improved behavioral phenotype as well in these individuals, in these mice. So this then, of course, then led to that indicated that they were on the right way with these metabolites. It was not only 4-EPS, but also several other metabolites that were linked to the microbiome. And so they then went to contract this specific project with us, a customized targeted panel that we developed using these specific aromatic microbial metabolites, specifically focusing on those and doing a quantitative customized assay on those. And they wanted it specifically for this clinical trial because their drug that they were using, this AB 2004, doesn't block any enzymes, it doesn't interfere with the biology. It actually absorbs aromatic metabolites. And so it was a intervention study that was focused specifically on these aromatic metabolites and seeing that it can bind those and then be excreted from the body. And so in the study showing that from baseline level to end of treatment, that there was a definite decrease in these aromatic microbial metabolites. And so the efficacy of the treatment was clear. And then also what they did see is that after stopping the treatment, these metabolites returned and there was an increase in these metabolites again. So really an effective tool to measure these and quantify the results at the end of the day. They also did a control and showed that in an acetylserine which shouldn't bind to that from a theoretical point of view, didn't bind, and so that was then confirmatory that it's very specific to these aromatic compounds. Then back to the efficacy, and so giving this treatment, they were then able to show that with reducing the levels of these microbial metabolites, the behavioral aspects, the irritability as well as behavioral symptoms reduced as well. So quite a nice correlation there between the metabolites and the phenotype. And then the GI symptoms that are usually associated were then also evidently treated because you had an improved performance of the GI-related problems in this cohort as well. So being able to measure these metabolites was really effective, and having this customized panel developed with them resulted in a very good end product for this client. So the study is really a nice example of combining, using firstly how important metabolomics was to identifying firstly the mechanism behind this, but then also supporting the drug development process. So global discovery was used to uncover these metabolites and the mechanism behind it. But then the customized panel that was developed quantified these metabolites quite nicely, and this was really able to help support them understand the mechanism of action quite a bit better. And so, of course, if this continues, this is one of many projects at the moment on autism. So if any of this develops further, it will be quite exciting to see that metabolomics was part of it and really being able to identify it further. So with that, brings me to the end, and I think that brings us to Q&A.